Okay, so welcome to this next video in which we are discussing clathrin-mediated endocytosis with a focus on Dynamin 1 and endophilin's role in this process. Okay, so we've now revised certain portions of the clathrin-mediated endocytosis pathway. We've revised the binding of this adapter protein complex 2 to the target protein's endocytic motif. Okay, we now want to discuss the recruitment of the protein clathrin to this complex, which is the protein that's actually going to cause the endocytosis, or at least the production of a bud, a vesicle bud on the membrane. Okay, so let's discuss the protein clathrin then. So, the protein clathrin. Clathrin is made up of two proteins, actually two polypeptides. Okay, so firstly, you have the clathrin heavy chain, which is what I've drawn here. And the clathrin heavy chain is often compared to the lower limb. So this is the clathrin heavy chain. Okay, and the reason it's compared to the lower limb is because effectively, this portion here is analogous to the thigh. Okay, this portion here is analogous to the leg. So remember, in anatomy, the leg does not actually mean what it means in popular culture. Most people would call the leg, most people would use the leg to denote what is actually called the lower limb. The lower limb is the thigh, the leg, and the foot. Strictly speaking, the leg is the portion where the calf muscles are. Okay, so it's the portion between the foot and the thigh. The portion where the tibia and fibia are. Okay. So, in fact, I'll draw this. If you've got a person here, okay, so here's their thigh, then they've got their kneecap there, and here's their leg, okay, and then their foot's there, and they've got some sort of swelling there. Okay, so this is the leg, strictly speaking, here, and this here is the thigh. So we're revising anatomy at the same time. So the whole thing is the lower limb, strictly speaking, but most people would use leg to mean lower limb. Okay, right. So, the clathrin heavy chain can be compared to the lower limb. So it has the thigh here, the leg here, and the leg slightly flexed on the thigh, and then the foot is horrendously plantar flexed, which means when you move your foot like in this direction, that's plantar flexion. Okay, so that's the analogy for what the clathrin protein's like. So this is clathrin heavy chain. Okay, and these portions actually have names. So this portion here, which was the analogous to the thigh, this is the proximal domain. Okay, so proximal domain. Okay, this portion over here, this is the distal domain. Okay, distal domain, so this is the portion that's analogous to the leg. And then finally, this portion here, which is analogous to the foot, its proper name is the N-terminal domain, because that's where the amino terminus of the polypeptide is. Okay, so the N-terminal domain. Right. Okay, so clathrin heavy chains also associate with a clathrin light chain. So the clathrin light chain associates with the proximal domain of the clathrin heavy chain. So here in purple is the clathrin heavy chain, and here in turquoise is the clathrin light chain. Okay, so there's the clathrin light chain. Now, basically, what you can do is clathrin doesn't usually exist like this in the cytoplasm. Instead, what you do is you assemble this into structures known as clathrin triskelions. Okay, so let me show you the structure of clathrin triskelion. Okay, so you have three clathrin light chains coming here. So here's one. Okay, so this is a clathrin light chain, clathrin heavy chain here. Okay, with its clathrin light chain. Here is another clathrin heavy chain down here with its clathrin light chain. Okay, here is another clathrin heavy chain here with its light chain. Okay, so that is the clathrin triskelion, this sort of bizarre swastika almost like symbol here. So this is a clathrin triskelion, what a clathrin triskelion looks like. Okay, and this is how the clathrin protein exists in the cytoplasm of the cell. Okay, so this is the clathrin heavy chain here, and this is the clathrin light chain here. So you have clathrin proteins, i.e. clathrin heavy chains with their light chains. They are assembled into these trimers, and this central bit here of the clathrin heavy chain, which is responsible for the trimerization, this is known as the trimerization domain. Trimerization domain. Okay, so each of the clathrin heavy chains contributes a portion in to bind the three clathrin heavy chains together in this trimerization domain. Okay, right. So what now happens is one of these clathrin triskelions is going to come and bind to our adapter protein here. And what's going to happen is that the clathrin is going to start polymerizing. Now let me show you how clathrin polymerizes. We'll do this in the next, not in the next video, on the next page. Okay, so how does clathrin polymerize? Basically, when it polymerizes, it forms buckyballs. It forms the same sort of structure as a football is. So it forms the shell of a football. So let me show you this. So the shell of a football is made up of pentagons and hexagons, all tessellating together. So let me have a go at drawing this. So here's a pentagon, okay, and we'll say this is the top of the clathrin complex, or the football. Okay, and now what's going to happen is you're going to get hexagons forming, well, joining on all around this, like so. Okay. And these hexagons are not going to be in the same plane as the pentagon. I've drawn the pentagon in the plane of the page, but the hexagons can't be in the same plane because it just doesn't work to put 5 hexagons around this pentagon. Instead, they're going into the page, they're folding into the page. So really this is a 3D structure that I'm trying to draw 2D. So imagine the pentagon is in the page. The hexagons are then folded into the page, and if you really want, you could actually cut out some regular hexagons and regular pentagons and try and build one of these things if you really need some intuition. Okay, and then all of these hexagons are sort of folding into the page, and then after that, even further folded in, there are these pentagons coming off these hexagons here. So 5-sided shapes come off here. So these are 2 sides. Then you have 1, 2, 3 more sides to make these 5-sided pentagons here. 1, 2, 3, 4, 5 there. 1, 2, 3, 4, 5 there. And again, whoops, 1 there, 1 there. Okay, so we're forming these pentagons attached here, and those will be folded round even more, and then you'll continue this process on. You'll put hexagons in here, like so, which you won't even be able to see really because they'll be folded round so much, and you'll continue the process on, and it forms a ball basically. And I'm sorry about my cut that I've just given you a fantastic view of. Okay, so it forms like a ball structure when it polymerizes. Now, how does clathrin form this structure? How do the clathrin triskelions form this structure? Well, let me show you. Let's highlight a clathrin triskelion here. So these are the 3 heavy chains. Then what we'll have is we'll have this one coming around here, and there's the foot of this first one. Okay, so there's a clathrin protein there. So if I add in the light chain will be here. Okay, so there's the light chain bound there. Okay, then off here comes another, whoops, need the highlighter. Here comes another heavy chain coming down here. And again, it will have its light chain coming off here. Okay, so here's its light chain. And then finally, the final heavy chain, and I don't want that, okay, is coming off down here, and here's its foot. And sorry, that's the final heavy chain. And then here's its light chain here. Okay, so there's a clathrin triskelion basically in this structure. Okay, and what's going to happen is these things don't perfectly make this up. You might hope that there's some beautiful way that these can all tessellate together so that they don't overlap and so that they perfectly make this buckyball structure, as it's called. And that's the fancy name for this. It's called a buckyball. Again, it's the structure that Buckminster fullerene, the carbon nanostructure has. Okay, you might hope that it perfectly tessellates without you having to ever have overlapping bits. That's not the case. You have overlapping portions of the clathrin triskelions. So, for instance, if we were to put another clathrin triskelion centered here, then this portion would be one of their a heavy chain, and it would go over this portion here. So let me show that. If we were to put another clathrin triskelion centered here, so these are the 3 heavy chains coming out like that, then its heavy chain will go off down here and overlap with this bit. So you will have overlap in the clathrin triskelions when you put them together like this. Okay, but overall they do form this buckyball-like structure. Okay, and that has this football circular shape. So, what's happening? What does this mean for the membrane? Because basically what we've just done is, if this is our plasma membrane here, we've just attached a clathrin triskelion underneath this membrane. Again, I'm not going to draw the full complex, I'm just going to draw the clathrin triskelion. Remember, it's attached to the adapter protein, which is attached to the synaptotagmin and also to the target protein. But here's our clathrin triskelion here. Okay, so I'll colour it in to make it look more familiar. Okay, so here's our clathrin triskelion. Okay, and now what's going to happen is it's going to start polymerizing. And when it polymerizes, what's it going to do? Well, it's going to form a ball structure here. So here's our clathrin buckyball starting to form. But this original clathrin triskelion that we had here was attached to the membrane. So what it's going to do is it's going to pull the membrane down with it, because that membrane is attached to this clathrin triskelion at the bottom here. Okay, so the membrane is a fluid, flexible structure, so it just gets pulled by this clathrin buckyball structure into a circular invagination, basically, and this is known as vesicle budding, basically. Okay, so we have budded off a vesicle from the plasma membrane, so this is vesicle budding. Right, okay, now, the clathrin buckyball, you might think, well, I know what will happen now. The clathrin buckyball will just continue on polymerizing, and it will create a full buckyball, and then it will just, chop off this membrane as it goes, so then you'll pinch off a vesicle. I see it all now. That's how it's going to work. No, not quite that simple. Instead, the clathrin polymer stops, basically. It just can't summon the energy, basically, to push this membrane inwards. It doesn't have the force required to push the membrane inwards there. So it meets too much resistance and it stalls. It stops polymerizing. So what we need now is some sort of proteins that are going to help us contract that neck of the membrane here, this neck of this budding vesicle, so that we can continue the polymerization of the clathrin buckyball and then finish this process and get a clathrin-coated vesicle. Okay, and this tucking in of the neck, this constriction of the neck, is what the function of the proteins Dynamin and Endophilin is. And we'll continue this discussion in the next video.